Now we discussed about the microsporogenesis and megasporogenesis. The formation of microspores from pollen mother cell through meiosis is called microsporogenesis. The process of megaspore from the megaspore mother cell is called megasporogenesis. Then we can discuss about the steps of microsporogenesis. Stamen consists of anther and filament. Anther contains 4 pollen sacs or microsporangia. Sporangium contains mass of large cells with prominent nuclei rich of cytoplasm. There are 3 types of walls in sporangia. Outermost single-layered epidermis, 2 or 3 layer of middle layer, that is endothecium, and innermost tapetum. Tapetum nourish the developing pollen grains. Some diploid 12 cells in pollen sac act as microspore mother cell. Meiosis or reduction division take place in microspore mother cell and form 4 haploid nuclei. At first stage, these 4 haploid nuclei bind together to form tetrad. At maturity, they separate and coming out from the anther. Next, microspore or pollen grain. The wall of microspore consists of 2 layers, outer tough exine and inner indane. A germ pore seen on side of pollen grain. First, the pollen grain nucleus haploid divide into a large vegetative cell and a small generative nucleus. The pollen grains are released by rupture of stomium along the line of divisions of anther and coming out. Pollen grain is regarded as male gametophyte because it produces male gamete or generative nucleus or generative cells. The vegetative cell degenerate, that is also haploid, and a single generative nuclei divide mitotically to form 2 haploid generative nucleus. Then we discussed about megasporogenesis. This is the structure of a typical ovary. Ovary with ovule. This is a single ovule. Here 2 integuments, nucellus, embryo sac. In embryo sac we can see 1 secondary nucleus haploid, middle haploid egg cell, 2 haploid synergids. Then this is the funicle attached to a fleshy part. Then the ovary contains 1 or more ovules, integumented megasporangia. Ovule develop on placenta. Here is the placenta, the funicle attached on the placental tissue. This is the single ovule. An ovule consists of a parenchymatous nucellus tissue with two coverings, that is integuments. A small passage, that is micropyle, opposite side of micropylar region is chalazal end. This is the chalazal end and this is the micropylar end. The stalk of ovule is called funicle. This is the funicle which attach ovule to placenta. Next, the steps of development of female gametophyte. Within the embryo sac, a large diploid cell, 2N cell, act as a megaspore mother cell. Meiosis takes place in megaspore diploid mother cell to form 4 haploid nucleus. 3 of them are degenerate and 1 haploid nucleus undergoes successive 3 mitotic division. 3 mitotic division to produce 8 haploid nuclei. 3 of them move to the chalazal end, that is antipodals haploids. 3 move into micropylar end, that is 2 synergid cells and 1 egg cell. 3 of them also are haploid cells. 2 haploid nuclei fuse together to form a diploid or 2N secondary or definite nucleus. Next topic, what is double fertilization and triple fusion? Here we can see double fertilization and triple fusion. At the time of fertilization, one male generative cell fuse with egg cell to form zygote. Another generative nucleus from pollen tube fuse with secondary cell to form triploid endosperm cell. This is the pollen tube, one of the generative nucleus fuse with egg cell to form diploid zygote, and another generative nucleus fuse with secondary diploid nucleus to form endosperm, that is triploid. This is the double fertilization. One of the generative nucleus from pollen tube fuse with egg cell to form diploid zygote, and one of the generative nucleus fuse with one secondary nucleus to form triploid endosperm. This is the double fertilization. Then what is triple fusion? Triple fusion means one of the generative nucleus haploid fuse with 2N secondary nucleus to form triploid endosperm, that is the triple fusion. This is the triple fusion and this is double fertilization.